The retina.
The retina is the innermost light-sensitive layer of the eye. It is composed almost entirely of cells which are arranged in layers or lamellae. The retina is a three-neuron system. The photoreceptor cells are the first layer. The second layer is composed of bipolar cells, amacrine cells, and horizontal cells. The photoreceptor axons synapse with the cells in the second layer. The bipolar cells synapse with the retinal ganglion cells in the third layer. The ganglion cell axons form the nerve fiber layer of the retina, which exits the eye through the lamina cribrosa at the optic disc to form the optic nerve. The ganglion cell axons extend to the lateral geniculate body in the thalamus of the brain. The giant glial cells of Mueller span the entire thickness of the retina and fill the spaces between the neurons. The internal limiting membrane is the basement membrane on the footplates of the Mueller cells. The layers of the posterior retina are shown in this photomicrograph. The inner layers include the internal limiting membrane, or basement membrane of the Mueller cells, the nerve fiber layer composed of ganglion cell axons, and the ganglion cell layer. Retinal layers located toward the vitreous are called inner. Layers located toward the sclera are called outer. The nuclear layers of the retina are composed of cell nuclei. They include the inner nuclear layer and the outer nuclear layer. The plexiform layers of the retina are composed of axons and dendrites. They include the inner plexiform layer and the outer plexiform layer. The photoreceptors are the outermost layer of the retina. Light has to pass through the entire thickness of the transparent retina to interact with the outer segments of the rods and cones. Parts of the photoreceptor cells include their axons or Henle fibers that comprise the outer part of the outer plexiform layer, the photoreceptor nuclei that form the outer nuclear layer, and the inner and outer segments that project through the array of intercellular connections called the external limiting membrane. The tips of the photoreceptor outer segments interdigitate with processes on the apical surface of the RPE cells. The RPE cells rest on the inner surface of Bruch's membrane. The choriocapillaris, the innermost layer of small choroidal blood vessels, is located on its outer side. The parts of a cone cell are seen in this scanning electron micrograph. They include the axon or Henle fiber, the nucleus, which is located in the outer part of the outer nuclear layer next to the external limiting membrane, and the inner and outer segments, which project through the external limiting membrane into the subretinal space. The photoreceptors, or rods and cones, are shown in this series of scanning electron micrographs. The parts of the photoreceptors that project through the external limiting membrane into the subretinal space include the inner segments and the outer segments. The inner segments of the rod photoreceptors are slender cylinders, while the inner segments of the cones have a conical configuration. The photoreceptor nuclei form the outer nuclear layer of the retina. The nuclei of the cones are located directly adjacent to the external limiting membrane. A red band highlights the external limiting membrane in this scanning micrograph. The external limiting membrane is a belt desmosome composed of leaky adherent junctions that join adjacent photoreceptors and Mueller cells. The inner and outer segments of the rods and cones are seen in this false colorized scanning electron micrograph. The photoreceptor nuclei form the outer nuclear layer, and the inner and outer segments project through the external limiting membrane. The cones in this scanning electron micrograph are colored red, blue, and green to emphasize that there are three types of cones that are sensitive to red, blue, and green wavelengths of light. Most of the outer segments in this photo are fractured and lost. The inner segments and the outer segments of the photoreceptors are seen in this photomicrograph. The inner segments have two components: the smaller myoid, located next to the external limiting membrane, and the larger ellipsoid, which is packed with mitochondria. The slender outer segments interdigitate with processes on the apical surface of the RPE cells. The photoreceptors blanket the outer surface of the retina, and their outer segments project into the subretinal space, shown here in the scanning electron micrograph. The junction between the inner and outer segments of the cone and rod photoreceptor are shown in these classic transmission electron micrograph from Hogan, Alvarado, and Wadel's textbook of ocular histology. The inner segments are tightly packed with mitochondria that provide energy for the photochemistry of vision. The mitochondria are found in the ellipsoid zone of the inner segments. A highly modified cilium connects the inner and outer segments. This connecting cilium has the 9+0 pattern of doublets seen in central nervous system cilia. The photochemistry of vision takes place in the outer segments of the photoreceptors. The outer segments contain a stack of discs composed of cell membranes. The visual pigment rhodopsin is present as a transmembrane protein in the cell wall of the discs. The arrangement of the discs in the outer segment has been likened to a stack of coins. A stack of Pringles potato chips is probably more appropriate due to their lipid content. The three membranes of the retina are shown in this diagram from Fine and Yanoff's textbook of ocular histology. The internal limiting membrane is highlighted in magenta. It is the basement membrane of the Mueller cells and is a true basement membrane. The middle limiting membrane is a linear band of synapses between the photoreceptor axons and the bipolar cells. It is located in the inner part of the outer plexiform layer. The external limiting membrane is not a true basement membrane. It is composed of intercellular connections called adherent junctions or zonuli adherentes that join adjacent photoreceptors and Mueller cells. In this scanning electron micrograph, the IOM has been peeled back, disclosing the retinal nerve fiber layer. This false colorized scanning electron micrograph of the interretinal layers shows the internal limiting membrane, the nerve fiber layer, a few cells of the retinal ganglion cell layer, and the inner plexiform layer. The internal limiting membrane is the basal lamina or basement membrane of the Mueller cells. It is the only one of the retinal membranes that is a true basement membrane. The IOM is secreted by and opposed to the basal surfaces or footplates of the Mueller cells. Extracellular matrix is attached to the basal lamina of the cells. The IOM serves as the attachment point for the fibrillar component of the vitreous humor, which is composed of fibrils of type 2 collagen. In this photomicrograph, the IOM is seen as a faint blue line on the basal surfaces of the Mueller cells on the inner surface of the retina. Mueller cell processes are seen separating bundles of ganglion cell axons in the nerve fiber layer. The red line highlights the location of the linear zone of synapses in the inner part of the outer plexiform layer, called the middle limiting membrane. Here, triangular cone pedicles and oval rod spherules synapse with the dendrites of bipolar cells and horizontal cells in the inner nuclear layer. Several cone axons or Henle fibers are visible. The photoreceptors project through the external limiting membrane. Only a few cones are present. The inner myoid and outer ellipsoid zones of the cone inner segments are visible. The rod spherules and cone pedicles are seen in this diagram of the middle limiting membrane from Hogan, Alvarado, and Wadel's textbook. In this photomicrograph, several capillaries of the deep capillary plexus are seen directly above the middle limiting membrane. The capillaries of the central retinal artery extend no deeper into the retina than the middle limiting membrane. The outer part of the retina is avascular and is nourished by the chorea capillaris. This false colorized electron micrograph shows the external limiting membrane of the retina, a belt desmosome composed of intercellular connections or zonuli adherentes that join the apices of adjacent photoreceptors in Mueller cells. The external limiting membrane is not a true basement membrane. It is also permeable or leaky. The inner and outer segments of the photoreceptors project through this belt desmosome like the cans of soda in this plastic six-pack yolk. Microvillus processes on the apical surface of the Mueller cells also project into the subretinal space. These are called the fiber basket of Schultze. To summarize, the layers of the retina are the internal limiting membrane, the nerve fiber layer, the ganglion cell layer, the inner plexiform layer, the inner nuclear layer, the outer plexiform layer, the outer nuclear layer, the external limiting membrane, the photoreceptors, and the retinal pigment epithelium. The RPE rests on Bruch's membrane on the inner surface of the choroid. A yellow spot called the macula lutea is present in the retina temporal to the optic nerve. This yellow spot is the basis for the commonly used but imprecise term macula applied to the posterior pole of the eye. The yellow pigment in the spot is derived from the diet. The area centralis, a more precise term, is applied to this area by anatomists. This refers to the part of the retina delimited by the superior and inferior temporal arcades of the retinal vessels, where the ganglion cell layer is thicker than a monolayer. The area centralis harbors the fovea or pit that is responsible for high-resolution vision. The fovea is about 1.5 millimeters in diameter, the same diameter as the optic nerve. The center of the fovea is located about 15 degrees temporal to the optic nerve. It is about 4 millimeters temporal to the center of the optic disc and about 0.8 millimeters below it. The foveola, or floor of the fovea, provides the sharpest vision. It is the only part of the retina that sees 20/20. The fovea of the same patient is imaged by spectral domain ocular coherence tomography and histology in this pair of photos. The layers in the OCT are generated by mathematical algorithms and do not correspond exactly with the layers seen in the histologic section. A single layer of outer retinal cells forms the floor of the fovea. The layer of outer retinal cells forming the floor of the fovea is composed of a tightly packed array of slender modified cones. The retinal pigment epithelium beneath the fovea is heavily pigmented. The ganglion cell layer of the perifoveal retina is quite thick, measuring 5 or 6 cells in thickness. In contrast, the ganglion cell layer outside the vascular arcades is an interrupted monolayer. The inner and outer plexiform layers of the retina appear quite different in this false colorized scanning electron micrograph. The inner plexiform layer is truly plexiform and has a spongy appearance. In contrast, most of the outer plexiform layer is composed of an array of photoreceptor axons or Henle fibers that are arranged in a parallel fashion. On either side of the fovea, the photoreceptor axons or Henle fibers are arranged in a radial fashion. The fovea would be located in the right of this photo. As noted previously, the deep plexus of capillaries derived from the central retinal artery penetrates only to the middle limiting membrane of the retina, which is not present in the fovea. This is responsible for the foveal avascular zone that is seen in this trypsin retinal digestion, or in fluorescein angiograms. The blood retinal barrier of the retina is shown in this photograph of the retina of an eye that was enucleated because it harbored a choroidal melanoma. The eye was removed immediately after the patient received an injection of fluorescein dye and was flash frozen and freeze dried. The inner part of the blood retinal barrier is composed of tight junctions that join the vascular endothelial cells. The tight junctions confine the fluorescein dye to the lumen of the vessels. The retinal pigment epithelium is the outer part of the blood-retinal barrier. The RPE cells are joined by an impermeable barrier of apical tight junctions. The yellow line is lipofuscin pigment in the basal cytoplasm of the RPE cells. Unlike the retinal capillaries, the capillaries of the choroid capillaris are fenestrated and leaky. The choroid is filled with green dye, which is confined by the RPE. The retinal pigment epithelium is largely responsible for the red-orange color of the fundus seen on ophthalmoscopy. In this macro photo, parts of the RPE have been removed, disclosing Bruch's membrane. Large vessels are seen in the underlying choroid through denuded Bruch's membrane, which is largely transparent. In this focally denuded specimen, some of the smaller choroid capillaries are visible, in addition to the larger choroidal vessels. The hexagonal mosaic of the RPE cells is seen in this flat preparation. The scanning electron micrograph shows the hexagonal RPE cells forming a monolayer on the inner surface of Bruch's membrane. Some of the cells are hidden by inner photoreceptor matrix material. The RPE is derived from the outer layer of the optic cup. The apices of the RPE cells project into the subretinal space, where apical processes partially envelop the tips of the photoreceptor outer segments. Large ellipsoidal granules of neuroepithelial melanin are concentrated in the apical cytoplasm of the RPE cells. The nuclei of the RPE cells are present in the basal cytoplasm, abutting the inner surface of Bruch's membrane. The shape of the RPE granules is reminiscent of mouse droppings. Granules of neuroepithelial melanin are always larger than the granules in the uveal melanocytes. The large ellipsoidal granules in the RPE cells at right contrast with the fine dust-like granules in the choroidal melanocytes in this flat preparation. The basal cytoplasm of the RPE cells contains lipofuscin pigment. This is visible as myriad autofluorescent golden yellow granules in this fluorescent micrograph. The lipofuscin pigment is produced by the continual phagocytosis and digestion of the tips of the photoreceptor outer segments by the RPE's phagolysosomal system. The lipofuscin pigment accumulates with increasing age. The RPE rests on the inner surface of Bruch's membrane, the inner layer of the choroid.